childhood trauma. It affects children's learning and their behavior, as in, we're in a classroom setting and we see a child and they're doing bad things and having temper tantrums, but on the inside, they're actually suffering from trauma, and the behaviors that we see are a result of that. And so, trauma informed practices utilize this knowledge that trauma affects learning and behavior to create practices that are sensitive to children's needs, like building positive student teacher relationships. And they can essentially ensure that all children can learn in the school environment, including those who've experienced trauma. So, what could be wrong with that? Well, the practices are currently being practiced within this one-size-fits-all approach, as in building that positive student-teacher relationship for one student looks the same for another. And yet if we tell a joke to one student and they're ha 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 laughing, but then we go to another student, they may be offended based on their own cultural background and lived experiences. And so that tells us that we need to use trauma-informed practices within a culturally responsive framework. Now, cultural responsiveness really takes this approach of thinking about children's lived experiences and their background. So they can essentially ensure that those same trauma-informed practices that we talked about can be implemented in a way that meets the needs of individual children, okay? And so trauma-informed schools and trauma-informed school programs need a little bit of help with their professional development. And so I've created a culturally responsive training program to implement within this trauma-informed school program. So we talk about things like historical trauma, racial trauma, inequities in schools, and the need for cultural responsiveness, as well as how to be culturally responsive. And so it helps to build up teachers' efficacy toward being culturally responsive, so yay. But more work is needed to make sure that teachers are able to really get from and interpret the content that's within these workshops. As in, we have teachers who may feel some discomfort with the content. Some may feel guilty or shame, and some may be uncomfortable with talking about race, which is essential to cultural responsiveness. And so future research and future work really needs to think about how we can best prepare teachers to engage in these conversations, as well as what can we do on our part to make sure that it is a safe and supportive environment. Essentially, these programs need to be culturally responsive and trauma-informed themselves so that teachers can go back and do the same for their students. Doing so will help to increase that cultural responsiveness, increase their ability to provide trauma-informed practices that meet the needs of students, and subsequently help our students to feel safe and thrive in the school environment. Thank you.
Osteoarthritis is a degenerative disease in the joint associated with aging and movement that can lead to severe pain. Almost 10% of people over the age of 60 suffer from OA. Maybe someone you know lives with osteoarthritis. For me, it's my grandmother. In horses, OA is naturally occurring, contributing to lameness in about 60% of horses, making them a perfect model to study and research osteoarthritis. The disease progresses when cells in the joint go into this inflammatory phase. Essentially, they stop producing all of those really good things like collagen, hyaluronic acid, the stuff that's in your face mask, and start producing the bad pro-inflammatory molecules. Currently, there is no cure, and the standards for treatment simply manage the problem, not improve it. Researchers found that if you take the elixir of life that is our blood and spin it down, you can concentrate all of the platelets with their great growth factors and host peptides and inject it right back into the joint to help assist with the healing process. This is called platelet-rich plasma, or PRP. My research mentor and team patented something called BioPly, which is a more refined version of PRP. And when tested against PRP, other treatments, steroids, and controls, the BioPly was able to downregulate the bad pro-inflammatory stuff and upregulate the good things like collagen and hyaluronic acid. Here's the roadblock. BioPly is not super easy to make. We require horse blood donors and have to spend about an entire day making it, which isn't bad, but what if we could find a way to make it more accessible? That's where I come in. Under the Schnabel Equine Sports Medicine Laboratory, my thesis focuses specifically on the individual peptides within BioPly that we've identified and what combination starts to create that desired BioPly effect. By researching this, we have the opportunity to synthetically make a peptide cocktail with all the beneficial peptides that could be distributed over the shelf, making it much easier than BioPly itself. So far, we've isolated some peptides, and the data is incredibly promising. And the tests start to rival the BioPly treatment. This research has the power to help entire horse populations. Not to mention, the treatments have translational effects into human medicine. Then maybe, who knows, someone in your life one day could be treated for their osteoarthritis with one of our famous peptide cocktails.
Thin film technology is important in every aspect of modern life, particularly in electronic devices and even in pasta dishes. But more on that later. These very thin layers of material have enabled tremendous advancements in technology over the last several decades. For example, consider a thin film coating on a battery electrode that enables longer lifetime in next generation electric vehicles, or creating a microchip for life saving medical equipment. And when I say thin film, I mean very thin, approximately 10,000 times thinner than just one strand of hair. The reason for such rapid improvement in these technologies is because we've understood more and more about how the films grow and how to control them in a specific application. This means, of course, that to continue advancing technology, we also need to deepen our understanding of thin film materials. In my PhD, I worked to understand at a very fundamental level polymer thin film materials. And I'd like to talk about this in terms of something easy to understand and that would make my Italian grandmother proud. You guessed it, pasta. When you're making a good pasta dish, you have to consider how each ingredient will interact to contribute towards the overall flavor and appearance of the dish. You can control the type of noodle, the seasonings in the sauce, and how long each ingredient simmers together. Similarly, in my research, I can make a recipe for a polymer thin film, where I seek to understand how new ingredients will affect the overall film. First, I introduce two new organic molecules. Think of this like a type of noodle. And from these experiments, I came to understand that film structure is critical in determining how fast a film will grow and what properties it will have. Now, speaking of properties, the next step was to introduce a new technique. This analyzes the mechanical resilience of the film without damaging the material. So you can compare this to a new tool to eat spaghetti that doesn't get the tomato sauce all over your clothes. These new materials and analysis that I developed during my PhD have provided important information to understand how polymers grow and how to use them in applications. For example, creating better batteries for a more sustainable future. More importantly, this research serves as a springboard towards overall improvements in understanding thin film materials, leading to enhancements in healthcare technology, transportation, and of course, better tasting pasta. Thank you very much for your time, and I hope you learned something about thin films.
When I was 15 years old, I learned through a high school genetics lab that the man on my birth certificate, my dad who raised me, was not my biological father, and he could never, ever know. My project, Ethics for Human Subjects, tells the story of misattributed parentage in the moment and aftermath of discovery. Over 30 million people have taken a DNA test. 2.5% of children are adopted, and somewhere between 4 and 10% of fathers are raising children who they mistakenly think are theirs. While my project is a novel, it is also in essence a case study of fractured identities, of the spectrum that lies between the innate desire to know one's biological family and the sincere reluctance to make those connections. On a craft level, I explore how mixed genre literature, performativity, and perspective can effectively represent the trauma that occurs in these moments of discovery through the telling of the story of three main characters. Katie Madrone, a fictionalized version of myself, is a precocious, science-minded outsider, a late bloomer, who writes letters and theatrical performances to cope with anxiety. Through interviewing family members and exploring artifacts, I was able to fill in the blank pages of a world of family secrets. Sasha Vale is based on interviews with the sister who I found at age 30. A well-grounded elementary school teacher in Portland, Oregon, she has always known of her adoptive status. A hefty folder of primary documents from the adoption agency and her biological parents helped me to tell her story. Finally, Kimbo St. Pierre, an actor and lighting technician in Olympia, Washington, does not become aware of being adopted until they take a DNA test at age 41, which also reveals an ethnic identity that it was not what they expected, sparking a journey for deeply buried roots. Kimbo is a composite character based on stories shared with me through a DNA Surprises group. Katie, Sasha, and Kimbo each grapple with what biology means in terms of their identities. Their stories tackle the following research questions. What catalysts tend to spark searches or refusals of genetic connections? How does the age of discovery impact the experience? What happens to relationships between family members who learn of a loss of genetic connection? What is the mental and physical toll of keeping secrets? And finally, how much of our being is tied to biology and how much is dependent on environment? Learning your genetic background is not what you expected can be a damaging and revelatory event for all parties involved. This project aims to contribute to the healing of families who struggle with such situations. Thank you.
Can you guess what is common in our laundry detergent and the James Webb Space Telescope? Yes, the use of nanomaterials. Materials having dimension in nanometers, approximately 100,000 times smaller than the diameter of a human hair. For their extraordinary properties, nanomaterials have very diverse use in cosmetics to therapeutics, in sports equipment to smart electronic devices, in environmental cleanup, and many more. The hype is because a tiny amount can tremendously improve materials' performance. On the other hand, there is a huge concern for nanotoxicity in terms of direct exposure to human and the environment. Some nanomaterials are non-toxic, like the zinc in our sunscreen, but most engineered nanomaterials have a certain degree of risk. Because of their teeny tiny size, they can easily interact with living cells and cause mutation or unknown damages. So, incorporating or embedding the nanomaterial in a safer matrix material like polymer or ceramic is a common practice that prevent the release of the nanomaterial in user environment and stop the nanopollution. But as the incorporation process is happening in bulk, there is a problem. The bulk matrix materials largely reduce the nanofunctionality. Imagine wrapping spaghettis with a blanket. Not seems very effective. Researchers all around the world are trying to find a better solution. Our research group works on utilizing the super strength, ultra low density, and excellent electrical signal processing properties of carbon nanotube, also called the CNTs. My research focused on strategic incorporation of CNTs in a safer polymer matrix to make a nanohybrid structure. I have developed a new method that coat the CNTs with safer nanolayer of polymer, like coating the spaghettis with olive oil, not with a blanket. This process is fast and well controllable. The safer polymer nanocoating not only keep the CNTs fully functional, but also help prevent the release of CNTs in dynamic use conditions. I have successfully demonstrated the use of this safe nanohybrid in 3 promising real-life applications: as a sensors in wearable e-textile devices, as a reusable super absorbent for oil spill cleanup, and in aerospace protection application. Whether we aim to build a colony on Mars or wanted to win the fight against cancer on Earth, the use of nanomaterial system is inevitable, and my research will contribute in developing a proper nanocoating method that will allow us to get the potential benefit from nanomaterials at a fullest extent effectively and safely.
Hello everyone! Today I'm going to be sharing with you all a very unique idea which can solve the problem faced by food processing industries and is costing them billions of dollars annually. So what is that problem? The problem is that the biofilms can grow inside the stainless steel equipments which are used to either process or treat the food. But what are biofilms? They are a group of microorganisms which can be pathogenic for human health consumption, and they can make our food unsafe. And if we eat that food, we can get really sick. So it is very vital for us to get rid of biofilms as soon as possible. They get enough moisture and nutrients from the food which is being treated inside those equipments. So currently in the food processing industries, chemical-based sanitizers are used to inactivate them. But there is one problem associated with using them, which is that those sanitizers can leave harmful chemical residues on the food which is inside those equipments. And those residues have shown to be carcinogenic for human health consumption. So what do we do then? I have a solution for you all, which is that instead of using these chemical-based sanitizers, we can use plasma-treated water, which is totally chemical-free. So what is plasma? Plasma is the fourth state of matter after solids, liquids and gases. And when we supply enough energy to third state of matter, we get plasma. So plasma is composed of powerful atoms, molecules and electrons. And when we treat the water with that plasma, all these powerful atoms, molecules and electrons get transferred to that water, and that water starts behaving as a sanitizer. There is one more advantage to using this plasma treated water, which is that it is totally an eco-friendly technique, because we were using compressed air as a working gas to generate plasma, suggesting that this plasma treated water can be used as an alternative disinfectant in the food processing industry to get rid of biofilms. Because one good news, when we treated the biofilms grown on stainless steel surfaces using plasma treated water, it was completely able to inactivate them. Thank you.
My name is Savannah Smith and I work with poop. If you're anything like my family, then when you think about poop, you maybe get grossed out. You just want it to be out of sight and out of mind. You definitely want me to stop talking about it. But when I think about it, I see so many potential resources in what some people think of as a waste. I want to take this unappreciated material and turn it into a source of power. If you live in a developed community, then when you flush your toilet, the wastewater most likely ends up in a wastewater treatment plant. Here, the water gets cleaned until it's good enough to be released back into the environment. But from this treatment, a large amount of solid material is usually left over. These solids are really difficult to deal with. They're heavy and they have to be trucked away. Best case scenario, we can use some of them to fertilize specific crops. But only the cleanest solids get to be fertilizer. More frequently, they just end up in a landfill somewhere. Wasting our waste contributes to greenhouse gas emissions, and it wastes a nutrient-rich resource that we could be using somewhere else. Now, you've probably heard that cow farts release methane into the air, which contributes to more greenhouse gases and then an increase in climate change. But what if we took those farts, captured them, and burnt them to create energy? This burning converts methane to carbon dioxide. And since carbon dioxide is a less harmful greenhouse gas than methane, this results in a decrease in the climate change potential of the gas by 1/30. Well, that's what I do with the solids from wastewater. If you take the solids left over from wastewater treatment and you put them in a sealed container, then you add microbes, which in this field we like to call bugs, the bugs will eat the solids and they produce methane gas. We can take this gas, burn it, and create power. The more the bugs eat, the more power we can generate. I'm trying to maximize this power by controlling which bugs are in the container and which are actually eating the foods. I do this by looking at the characteristics of the bugs that are already in the container and ones that could potentially get into the container with the poop. From this information, we can adjust certain parameters, like the amount of food or the temperature, and encourage certain bugs to eat and grow. If done correctly, we can reduce the amount of solids that we throw away and create a reliable energy source out of something that we would otherwise be throwing away. By doing this, we not only reduce the amount of waste that goes into landfills, but we're actually creating a power source out of a material that's readily available in many areas and is often discarded. Methane is an energy-rich power source that can be used in a variety of applications. So this is a potential solution to global waste issues, greenhouse gas emissions, energy demands, and ultimately, climate change and the health of our environment. Let's turn some more poop into power.
Remember the recent fuel shortage in North Carolina and the panic that ensued? Incidents like this are a wake-up call that demands for the development of alternative sources of energy. For example, exploring microorganisms' natural ability to produce biofuels. My research contributes to the solution of this problem by introducing a bacterium from Clostridium species. It is nurtured in a special chamber filled with nitrogen and hydrogen gases and looks like small rods under a microscope. This microorganism has innate ability to eat sugars and convert them into industrial chemicals such as butanol. Butanol has ideal chemical and physical properties to be used as a transportation fuel and has many applications in textile, construction, and cosmetic industries. The demand for butanol is only going to increase in the days to come. According to an estimate, the global market size for butanol will reach $4 billion by the year 2025. My goal is to meet this rising demand by channeling the abilities of Clostridium. There is, however, a catch. Working with this microorganism is not easy. It needs expensive laboratory equipment to grow in an environment lacking oxygen, and the quantities of butanol produced are not sufficient for use on a large scale. In order to address these bottlenecks, I have developed a method to genetically modify this organism. The modification will allow us to grow this bacteria aerobically, simplifying the experimental conditions and saving a lot of costs. I've also identified prospective genetic targets that can be manipulated to improve this microbe's capacity to produce butanol. It gives us the opportunity to develop an industrially suitable version of this microorganism. My findings will also open the doors for future researchers to meet the ever-increasing energy demands. When it comes to the potential of microorganisms, there is still a lot to explore. Many known and unknown organisms possess unique traits for enabling the development of biotechnologies to produce fuels and commodity chemicals. Not only this, these special creatures can save us from the devastating impacts of climate change. Along with wind, solar, and tidal energy, microorganisms can help us reduce our carbon footprint. These little creatures, isolated from soil, quite literally be the powerhouses of the future.
Hello, everyone. My name is Juan. I'm a PhD student in food engineering. Today, I want to talk about our research called Plasma Technologies for Hydroponic Seed Basic Growth, which can be produced on location and on demand. But what is plasma? Well, plasma is the fourth state of matter, and we have seen it in nature, in the northern lights, and in lightning. You see, when you have a gas, such as air, and you add enough energy to it, it can transition into a plasma phase, where it contains reactive nitrogen and oxygen species, such as nitrate. This component, nitrate, is relevant because of the commercial fertilizer industry, which uses it in their formulations to grow plants. However, in contrast to commercial fertilizers, plasma can be produced both at atmospheric pressure and at colder temperatures, such that if you have a plasma source and expose it to a liquid, we can transfer the nitrates to that liquid and possibly use it to grow plants. This is relevant because of the $9.5 billion hydroponic industry, which uses a liquid called nutrient solution to grow plants without soil, as you can see in the picture on the left. Since this liquid is recirculated throughout the whole system, if it becomes contaminated by microorganisms harmful to plant health or to human health, then the hydroponic farmer may lose their whole crop. And if they didn't catch the contamination on time, then their produce can be a vector for foodborne illness spread. An example of this is sweet basil, which as early as February of this year was involved in a recall due to the risk of foodborne illness to the consumer. So we knew we wanted to help hydroponic farmers, and we knew from the literature that plasma could inactivate microorganisms or could help with plant growth. But we saw a gap in terms of plant quality. So that's what we did. We look at the plant growth, the plant quality, and the sanitation of the growth environment of control sweet basil plants compared to sweet basil plants grown in plasma treated nutrient solution. Our results showed that we used a similar amount of resources in terms of land and energy and water. But looking at the plant quality and plant growth, our plants were more attractive because they were taller and they had more leaves. This is especially exciting because conventional sanitizers in the hydroponic industry can harm plant health, but in our case, we actually improved it. Furthermore, when we looked at the sanitation of the growth environment, we saw that plasma was better at inactivating microorganisms such as algae and non-pathogenic E. coli compared to the control. Therefore, we concluded that plasma is a promising alternative for hydroponic farmers to grow both safer and higher quality produce. Thank you for listening. And also, I would like to thank our collaborators at Drexel, Rutgers, and the USD and IFA, without whom this study could not have happened.
Every day, 17 people die in this country while waiting to get an organ transplant. Every 9 minutes, another person is being added to that waiting list. So how can we solve this crisis? Clearly, waiting for someone else to donate the organ isn't working. An effective alternative would be fabricating those organs. Using 3D printing and other revolutionary methods, we are on the verge of developing human organs in laboratories. Did you know all the organs in our body generated from a single cell type? These are called the stem cells. So if you want to develop an organ for a patient, extracting his stem cells and converting them to organ-specific cells is the way to go. Suppose the patient needs a new heart. You collected his stem cells, added them to the growing heart, and followed all the protocols to convert them to heart cells. Now, how do you know that the cells are alive deep inside that new heart and if they have converted? You cannot just slice the heart in half to check, right? Because then you don't have an intact heart anymore for the patient. That's where my research comes in. I am developing an artificial intelligence or AI-based approach to check the presence and conversion of stem cells. As long as the cells are alive within the heart, their presence will be captured by a sensor that measures the electrical properties from outside without disturbing the organ. Now, knowing that the cells are alive, the next question is, have they converted into heart cells? I observe in each stage of the stem cell conversion, the electrical properties change a bit. This results in change in signal pattern. So in our case, the patient stem cells would show one kind of pattern, while the converted heart cells would show a slightly different pattern. It's very difficult for humans to track those changes, but my proposed AI approach detects them with very high accuracy. Just imagine the possibilities it represents. We can control the organ growth precisely while developing multiple organs for multiple patients in parallel. I strongly believe my research finding will play a significant part in ending the organ shortage forever. Thank you.